Hi, welcome to my channel. In this video, I will show you how can we perform immunofluorescence experiment in the lab. First of all, what is immunofluorescence experiment? It is a technique used to visualize the location of proteins in cells or tissue sections using fluorescently labeled antibodies. In a simple way, immuno means antibodies and fluorescence means fluorescent dye. So how does this technique work? For example, I have a tissue section where I want to detect a target protein. To detect this protein, we will add primary antibodies that will specifically bind to that protein. Then, we will add secondary antibodies that will bind to primary antibodies. Also, these secondary antibodies will be labeled with a dye that will give us signals and we will detect our protein. So let's go through the protocol step by step. Also, you can ask me any question and I will try to explain and help. The first step will be baking your slides. So first of all, you will be having FFPE slides, which are formalin fixed paraffin embedded tissue slides. So the first step will be to bake the slides at high temperature. It is very important step because baking will make your tissue section strongly attached to the glass slide and it will not detach during the whole experiment. Also, this step will help to melt the paraffin so it will easily come off during the next washing step. So we will put our slides in slides rack and then the rack can be of glass or steel, both works fine. Then we will bake the slides in an incubator for 90 minutes at 60 degrees. After baking, you will take your slides out of the incubator and label them. For example, you will be having one slide on which you want to check expression of your desirable protein and one more slide to use as a negative control. So negative control is basically a slide where we don't put any antibodies. It is just to confirm non-specific binding of primary antibodies as well as secondary antibodies. Also to check if there is any autofluorescence, which is a natural emission of fluorescence by biological structures or molecules without any fluorescent dye. Also, always label your slides with pencils because pencil will never come off with xylene and ethanol wash. The next step will be removing wax from your slides. So we will put our slides back into the rack and wash with xylene and ethanol. So first of all, we will wash our slides with xylene for 5 minutes. Once again, we will wash with xylene for 5 minutes. And now to remove xylene from our slides, we will wash the slides with 100% ethanol. And finally, we will give one more ethanol wash to completely remove xylene from our slides for 5 minutes. After completing the washing with xylene and ethanol, finally we will put our slides in a glass bucket full of tap water and then wash the slides with running tap water for 10 minutes. The next step will be antigen retrieval. This step is important because you need to bring your antigen that you are targeting, or you can say your desirable protein, to the surface so the antibodies can easily attach to it. So after washing slides under running water, we will put the slides in a plastic bucket with 1x EDTA buffer and incubate for 30 minutes in a microwave oven with maximum temperature. Also, don't forget to remove the steel handle from tissue rack because after incubating, it will be super hot. After incubating with 3 EDTA buffer, we will again wash the slides under running tap water for 10 minutes. So the next step will be making hydrophobic barrier around the tissue. After washing, we will create a hydrophobic barrier around the tissue on slide using image pen. This pen is basically a wax pen and the purpose of this step is to avoid antibody solutions and other reagents spreading across the slide. After making the barrier, we will place the slides in slide box and let the wax get dry on the slides. During that, add few drops of water on the tissue so the tissue won't get dried. After the wax gets dried, we will wash slides in 1x TBST for 5 minutes. During that, when the wax on our slides are getting dry, we will prepare blocking solution which will be 10% normal gauge serum. Blocking solution is important as it will prevent any unspecific binding of antibodies on tissue. For example, if your target proteins are at specific location on the tissue, the remaining area will be covered by proteins present in blocking solution, and your antibodies won't bind to those areas as your antibodies are not specific for those proteins. During that time, we will also prepare primary antibodies according to manufacturer instructions. Usually the dilution is 1 in 1000. Now we will put blocking solution and incubate the slides for 1 hour. After that, we will remove the blocking solution and add primary antibodies and incubate the slides overnight at 4 degrees. So the next step is adding secondary antibodies. Next day, we will remove primary antibodies and wash slides 3 times with TBST. Add secondary antibodies and incubate for 1 hour. After 1 hour, we will remove secondary antibodies, wash again 3 times with TBST and add Sudan black solution to avoid autofluorescence, and we will incubate the slides for 30 minutes. After 30 minutes, we will wash the slides again with TBST 3 times. The final step is sealing the slides. Now we will seal our slides using mounting media. Usually we add 150 microliter of mounting media and then we will put cover slips carefully to avoid making any bubbles on tissue section. Finally, we will seal the slides using nail polish to properly seal the slides and avoiding any leakage of mounting media. Now your slides are ready to image, but if you are not imaging them, you can keep them at 4 degrees. Thank you for watching. In next video, we can talk about the role of different chemicals and how we can prepare them. Like and subscribe if you like my video, and I'll see you in next video. Thank you so much, and don't forget to ask any question if you have, and I will try to answer and help.